Okay, so ladies and gentlemen, you are welcome. So I think if you could remember, okay, good afternoon, Oga Uloma. So you are welcome. So please, what are we going to do? Are we going to answer ACO objectives? That is the first thing that we need to agree on. Are we going to answer ACO objectives questions? Because of course, with me, I have questions on enzyme. Sorry, I have some objectives questions, and I also have essay questions. So which one are we going to answer? Okay, so someone said both. Someone is suggesting both. So I think if it is objectives, so let's start with the objectives and then briefly we move to the essay. So let me share some questions here with me. Let me share some of the questions. Okay, someone said essay before objectives. Okay, so let's start with the essay. Do you want me to use both, or you want me to answer it through interaction? Okay, I think let me use the board. That would be, since you said you prepare to start the AC. So let's see. Questions that we started answering last time. So you see, the first question says, using a specific example, briefly highlights the relevance of chemistry to understanding the key concept in nursing. That is the first question. And then the next one is the ABO blood group system is related to some glycoproteins. We discussed on that. Then sequentially list and describe the 6 classes of enzymes. We answered this. What is the relevance of reactions in our body system? So if you could remember vividly last time, question 1 to D, we got this last time. So if you want to get the answer to these questions, if you are absent last time, I will send you a link on the solution to question 1D. So we have a complete video on that. So now we are going to start with question 2 today. So I think for those of you that attended our last discussion, you will of course you will believe me that we have actually discussed question 1 to D. Do you agree that last time we discussed or we answered question 1 to D? If you are there, you attended the lecture from question 1 to D, say hi. So for today, we are going to answer from question 2 to question 6. So if you attended the last class, say hi. The last time we answered question 1 to D, say hi. Okay. So thank you, ladies and gentlemen. You see the first question 2A. We are now going to start with question 2A. This is biochem 241. So we are going to start answering question 2a. So you see, question 2a stated that, okay, someone is asking that why are you saying hi to? I said last time I answered question 1a, b, c, and d in one of the revision class that we did. So today we are going to answer from question 1 to 6. Attended, or if you watch the video on the last revision, you say hi. That is what I said. So for the 2a, you see the 2a says highlight the relevance of Human Genome Project to medical biochemistry. Highlights, highlights, highlights the relevance of Human Genome Project. Highlight the relevance of Human Genome Project in medical biochemistry, in medical biochemistry. So that is the question. So you see, so we are now going to start with question 2a. What is the relevance of Human Genome Project in biochemistry? So that is where we are going to stop. That is where we are going to start, ladies and gentlemen. But you need to understand the meaning of Human Genome Project first. And I think if you are watching some of my videos, you will see where I actually discuss the Human Genome Project. Yeah, Cynthia, I will send the link for question 1 to D that we answered last time. But now let's concentrate on 2A. So now, what is Human Genome Project? A Human Genome Project is a project that was conducted by scientists. So ladies and gentlemen, generally, before the Human Genome Project, there is a lot of mysteries. There are a lot of hidden information in relevant or in relation to the biochemistry. In fact, not only biochemistry, in the medical sciences in general. So it's after the Human Genome Project that a lot of information revealed, a lot of information came out, and that is when scientists was able to come up with a good concept of what exactly happened in medicine. So what is Human Genome Project? A Human Genome Project is when the complete DNA of human living organisms was sequenced. And the project was started in 1990. So the Human Genome Project started in 1990 and end in 2003. So if you can see, to sequence the whole, because the word when we say genome, ladies and gentlemen, the genome means is the complete, is a complete genetic makeup, is a complete genetic makeup of a living organisms. That is what we mean with genome. The first thing, a genome is the complete genetic makeup of a living organisms. So when we say genetics makeup or a complete setup of a genetics, or sorry, a complete set of the complete genetic makeup of a living organisms, it means DNA. So the DNA of a particular living organism is their genome. And ladies and gentlemen, that is why we said that DNA is a language of life. If you are beautiful, if you are intelligent, if you are dull, if you are ugly, it's all because of your DNA. So I'm the way I am because of my DNA. The intelligence in you, the way you talk, the shortness or tallness of a particular living organism is because of their own DNA. So that is where we said genome, it means the complete set of a genetic makeup of the living organisms is what we call genome. So that genome means DNA. Like for example, if you can now get the DNA of Mukhtar Ali, you can get another Mukhtar Ali. So if you want to produce me, you can get my DNA and get me. If you want to understand the behavior of Mukhtar Ali, you can use the DNA. So now, the DNA means genome. That is the simpler way. So we said human genome. What does that mean? It means the DNA in human. A project, so human genome project, it means it's a project conducted by a scientist in order to come up with the information, the languages, the letters that make up our own DNA. Remember, ladies and gentlemen, what are the monomers of DNA, please? I want to get this information. What are the monomers of the DNA? Please write it on the comment box. The monomers of DNA are called what? What are the monomers of DNA? The monomers of DNA are called what? What and what? What are the building blocks of DNA? Please, I'm waiting for your response. What are the monomers of? No. What are the monomers? Please, this is one of the most basic things that you should know. What and what join together to produce DNA? Please write it on the comment box. Kai, no. Excellent, Simon. It's nucleotides. So nucleotides are the molecules or are the monomers. We call them monomers of DNA. So it is these nucleotides that join together to produce DNA. So now the question is, what types of bonds that, like for example, what I mean here, the nucleotides are the small molecules like these. There are small molecules like these that join together. So these are the nucleotides here, these are the nucleotides, and then this is the DNA. And remember, ladies and gentlemen, DNA is a double helix. So this is the DNA. So therefore, the nucleotides join to produce DNA. So this is the DNA, deoxyribonucleic acids. So now I have another questions. What is the names of bonds that join the nucleotides together to produce the double helix DNA molecule? Please, I need answer to this. What is the names of bond that linked the nucleotides together to produce DNA? I need answer to this. The name of bonds that join the nucleotide together to form the DNA is called what? No. It's not excellent. Taima. The bonds joined together to produce a DNA molecule is called phosphodiester bond. You see, ladies and gentlemen, I always while answering the question, you see, I realize that many of you are not here. Many of you are not here. And I said that I will take attendance. So I'm seeing you. So what I'm seeing here, we have nucleotides. So these nucleotides, they are the blocks. They are like blocks that are used to construct DNA. So how do they construct? So they use a pause to join themselves. They use a bond to join themselves. And the force that join these nucleotides together is called phosphodiester bonds. So I always, when answering question paper, I always try to provide explanations so that at least if you are not given the same question, but you will have an idea on how to answer some of the questions. So therefore now the nucleotides join together through phosphodiester bond to produce DNA. And one very interesting thing, ladies and gentlemen, you should know that the DNA is double helix. It's double helix. So when you say it is double helix, it means that it is made up by two strands. Two strands. They are joined by phosphodiester bond, and this DNA is double helix. That is, it's made up of two strands that join themselves complementary. So now I have another question, ladies and gentlemen. What is the name of bond that join the two strands together? What is the name of bond that joined or that stabilize the double or the two strands together to make a double helix DNA molecule? Please, I'm waiting for your answer. What is the name of bond that stabilize or that join the two strands of the DNA together? No, it's not phosphodiester bond. So a phosphodiester bond is the bond that join the nucleotide together. So after you join the nucleotide together, you have one strand like this and you have another strand like this. That makes the double helix DNA. That is made up of two strands. So what force or what types of bond that joined the nucleotides? So that join the two strands of the DNA together. That is the question. Excellent, excellent. Aisha Obala Barbara. The answer is hydrogen bonding. Hydrogen bonding. So it is hydrogen bonding that join the two strands of the DNA together. So hydrogen bond stabilize the double helix of the DNA molecules. And how do we read the DNA? We read DNA. This is how it is. We read it from 5' to 3'. And then here, so this is what we call the first strand here, and then the other one is reading from 3' to 5'. So you see there is a pairing between the nucleotide. So guanine always pair with cytosine, and then adenine pair with thymine. So this is how it is. You have guanine also here pair with another cytosine. Cytosine pair with guanine. Then you have thymine pair with adenine. So this is how the double helix of the DNA formed. And very importantly, ladies and gentlemen, this is what we call a Chargaff's rule. So you shouldn't also forget about this. We have what we call Chargaff's rule. So a Chargaff's rule is a rule that trying to tell you that a Chargaff's rule is a rule that trying to tell you how the base pair with one another. So therefore, guanine usually pair with cytosine through 3 hydrogen bonding. So between guanine and cytosine, there are 3 hydrogen bonding. And then between adenine and thymine, there are 2 hydrogen bonding. So guanine pair with cytosine, and then adenine pair with thymine. So let's say, ladies and gentlemen, in a DNA molecule, let's say you have 256 guanine. and then you have, let's say, 200 adenine. So if you are asked to define the number of nucleotides that are found in that DNA molecules, please can you tell me the number of the nucleotide? I think it's simple. Well, let me start by this. If you have 256 guanine in the DNA molecules, how many cytosine do we have? If you have 256 guanine in the DNA, how many cytosine do we have? I need this answer. If in the DNA molecules you have 256 guanine nucleotides, how many cytosine do we have? If you have 256, please, I need answer. Urgent, please, you should be passed. If you have 256 guanine, how many cytosine do we have in the DNA molecule? Someone said 0. Excellent. Is it Osong? Because the answer is 256. Because guanine is always pairing itself with cytosine. So therefore, if you have 256, it tells you that there is also equal amount of cytosine. So the answer is 256 cytosine. So that is the answer. So now, ladies and gentlemen, this is very important. So which of the rules trying to explain this rule is Chargaff's rule? So a Chargaff's rule is a rule that states that in a double helix structure DNA, guanine always pair with cytosine and adenine always pair with thymine. So the number of hydrogen bond between a pairing of adenine and thymine is 2, and then the number of hydrogen bonding between guanine and cytosine are 3. So you shouldn't also forget this pairing explanation. So after this Chargaff's rule, so we are now talking about the nucleotides, Human Genome Project. So a Human Genome Project is a project that was conducted by scientists in 5 different countries. Because the human genome is very large, so the scientists cannot be able to sequence the whole of the DNA in one country. In fact, in one lab. So that is why they have to break down the DNA molecules into 5 fragments. They have to cut the DNA into 5 fragments. Some of the DNA was taken to China, some to Germany, some to USA, some to Japan, and I think the other one is in UK. So these are the 5 countries where they sequence the Human Genome Project, and they started in 1990 and they completed the project in 2003. So after the Human Genome Project, it means that what the scientists did was that they get the DNA of human and then they sequence it. When we said they sequence it, it means that they determine the arrangement and the number of nucleotides that are found in the DNA. And that is how they are able to reveal that in the human genome, there are about 3 times 10 raised to the power of 9 base pairs. So when we say base pairs, it means nucleotide. So in the human genome, there are about 3 billion, the 3 times 10 to the power of 9 means 3 billion base pairs. So the base pairs means nucleotides. So there are about 3 billion base pairs in human DNA. That is how this. So that is how the scientist was able to know that in the human genome there are about 3 billion base pairs. That is 3 billion nucleotides in human DNA. And very importantly, ladies and gentlemen, the size of the DNA varies across organisms. So the number of nucleotides or the number of base pairs that are found in human DNA is different from the number of nucleotides that are found in bacterial DNA. Is different from the size as well as the number and the arrangement of the nucleotide sequence that are found in sheep, in goats, in donkey, in monkey. So it is because of this variation in terms of the number and the size of the nucleotides across organisms that is where we are different from them. So the more close we are, the more closeness between the human genome and the genome of other organisms, the more close they are in terms of the number and the size as well as the arrangement of the nucleotides, the more we look alike. So that is why if you look at human and you look at chimpanzee, you see that we are similar. We look alike with human and chimpanzee. Bukola, thank you very much for joining the YouTube membership. So if you are a first semester student, I advise you to always join PQ and short videos because that is where most of your videos are. So if you join level 1, you need to actually upgrade yourself. And don't forget that for those of you that are just coming to the YouTube channel, actually the channel, we have videos on biochemistry, we have videos on biostatistics, videos on human physiology 225, human physiology 227. We also have solutions to past question papers as well as the courses or the second semester courses. We have all the videos here. But most of those videos, especially biostatistics and human physiology, if you are not a member, you cannot watch them. So if you want to join the YouTube membership, click on the join button and then you use your ATM and join. So you'll have access to all our videos. So now, after this human genome project conducted by the scientists across 5 countries started in 1990 to 2003, that is when the number of genes in humans are determined. So like in humans, there are about 25,000 genes. And these genes, ladies and gentlemen, is what actually make us the way we are. Like for example, the genes, like for example in humans, we have hemoglobin gene, that is HB gene. So hemoglobin gene is a gene that is responsible for producing hemoglobin. We have opsin gene, What is opsin gene? Opsin gene is a gene that produces opsin protein. And opsin protein is a protein that is important for our vision. That is our seeing. So we see with our eyes because of this opsin. Some child are given or are born with a defect or with a problem in opsin protein. That is why they cannot see. That is why they will have or they have a vision problem. We also have FOXP2 gene. A FOXP2 is a gene that is found in humans that allow us to talk. So ladies and gentlemen, so what are the relevance now of human genome projects in medical biochemistry? Because that is the question that we are going to answer. So ladies and gentlemen, go and ask, go and check, go and confirm when was the knowledge of sickle cell disease revealed? When was the scientist was able to know more about sickle? Is after the Human Genome Project. Because before, nobody will tell you this is what sickle is all about. So it's after the complete Human Genome Project that is when different genes in humans are known, after the Human Genome Project. So now, what are the problems the scientist was able to see after revealing the Human Genome Project, especially related to sickle? So hemoglobin is a gene that codes for hemoglobin as a protein. And this hemoglobin protein, it is tetramer. It is tetramer. It's made up of 2 alpha chain and 2 beta chain. And the alpha chain is made up of 141 amino acids. And the beta chain is made up of 146 amino acids. So 2 alpha chain means that you have 141 times 2. And this will give us 282. And then, of course, 2 beta chains will give us 146 times 2. And this will give us 292. So therefore, you can add this, and this will give you a total number of amino acids that are found, because hemoglobin is a protein, so protein are made up of an amino acid. So that is when the scientist was able to know the number of nucleotides that code for hemoglobin gene, the number of amino acids that are found in the hemoglobin protein. And that is when they are able to see that, okay, in the hemoglobin gene, is just a substitution. It's just a substitution, that is mutation, replacing thymine with adenine, and that will lead to coding of an amino acid called valine instead of glutamic acid. Because, of course, in the hemoglobin gene, it has 146 amino acids. So this is like how the amino acids arranged in the hemoglobin. So they arrange like this. We have like about 146 amino acids in it. So you see, after the scientist was able to, after the complete Human Genome Project, the scientist was able to know that, okay, 1, 2, 3, 4, 5, 6. At the position 6 of this hemoglobin protein, the amino acid that is supposed to be there is glutamic acid. But because of this mutation, replacing or substituting adenine with thymine, instead of glutamic acid to be at position 6 of the beta hemoglobin, it now ends up coding amino acid valine. So therefore, the story, the narration, the concept behind sickle cell anemia is just the change of glutamic acid with valine at position 6 of beta hemoglobin gene. That is what changed the narrations of those individuals with sickle. And of course, ladies and gentlemen, you see, all this revision that we are doing, I'm just trying to make sure that before we answer this question from beginning to the end, we will actually discuss the whole of the course. Because of course, if you check in your course, where you will see where the issue of the substitution of the sickle cell disease. So you see, ladies and gentlemen, I think even from this explanation, you will now understand that with the knowledge of Human Genome Project, it can help in the applications, or one of how it's actually linked to medical biochemistry, is disease diagnosis. Diseases diagnosis. So with the knowledge of Human Genome Project, it can help scientists to use or to isolate a DNA and determine or check a gene to see whether it is actually related with some, or with the presence or absence of a disease. So that is why one of the relevance of Human Genome Project, you can use it in disease diagnosis. So it helps in disease diagnosis. And then number two, the knowledge of Human Genome Project is actually relevant to medical biochemistry in terms of disease, or let me say drug development. Drug development. Apart from drug development, also vaccine development. So with the knowledge of Human Genome Project, in fact, it's after this Human Genome Project that a lot of scientists was able to come up with an idea of how to discover new drugs, how to come up with new vaccine. Because the vaccine that we are talking about, we have different types of vaccine. We have protein vaccine, we have attenuated vaccine, we have DNA vaccine, we also have RNA vaccine. So the development of DNA and RNA vaccine is after the complete Human Genome Project. So you see now, the relevance of Human Genome Project in medical biochemistry, it aids in disease diagnosis, drug development, vaccine development, disease treatment. You can add with that. Number four, disease treatments. So, ladies and gentlemen, did I do justice to this question? Did I do justice to this question, please? Let me show you the question again. Did I do justice to the question? So you see, the question says highlight the relevance of human genome to medical biochemistry. So you see, it's two questions. If you can just mention these four points, you'll get your full marks. Disease diagnosis, drug development, vaccine development, and all this. Sorry for the inconvenience. So it's actually a network problem. It is a network problem. So can you hear me now? Can you hear me now? Okay. So now the first question here is, we are to highlight the functions of the following. So what is the functions of smooth endoplasmic reticulum? So the major functions of smooth endoplasmic reticulum is lipid synthesis, lipids biosynthesis. So it is organelles of the cell where a lipid can be produced, that is number one. And then apart from lipid biosynthesis and also drug metabolism. So these are two major functions of smooth endoplasmic reticulum of a cell. So one of these functions is involved in protein synthesis, that is production of proteins, and the number two, drugs metabolism. So any drugs that we take, the drugs is metabolized in the liver as a hepatocyte. Hepatocyte, it is a cell of the liver. So in these hepatocytes, there are organelles, of course. There are organelles. So when you say organelles, there are organelles. So the organelles which is in the cells of the hepatocyte include smooth endoplasmic reticulum. So anything that we take, we transport it. After we take it into our systematic circulation from the blood, it needs to be transported to the liver, and from the liver now will use the different types of the mechanisms of transportation to take it into the cell. Like for example, we have different types of transport of material across the cell. We have facilitated diffusion, we have simple diffusion, or we can set passive transport. Passive transport. And then we have active transport. We have active transport. And then number 3, we have bulk transport. These are different types of transportation of material across the cell membrane. And each of these, they have their own types of transportation. Sorry, they have their own division. Like for example, passive transports. In passive transports, we have simple diffusion. We have facilitated diffusion. We have facilitated diffusion. Then we have osmosis. These are the different types of transportation of material. So for the passive transport is divided into 2. Then for the active transport, we have primary active transports. We have secondary active transport. And then, of course, for the bulk transport, is divided into number 1, we have endocytosis, and then we have exocytosis. And then, of course, for the facilitated diffusion, it is also divided into 3. We have antipod, we have sympod, we have unipod. And then these are the different types of diffusion that we have. Please, is it lipid biosynthesis or protein synthesis? All the two. It can synthesize protein, it can synthesize. No, protein synthesis is not even smooth endoplasmic reticulum. Proteins are synthesized in the ribosome. So ladies and gentlemen, these are the different types of transportation. So what we are saying, drugs is metabolized in the hepatocyte, specifically in the endoplasmic reticulum. So when you take drugs, the drugs will actually go into our systematic circulation. So after systematic circulation, then it needs to be transported to our different organs. So to our different, in the blood circulation, it needs to be transported to the liver. So from the liver, because liver has a cell, so the drugs must find its way to get into the cell itself for it to reach the smooth endoplasmic reticulum. And then so you have different types of drug transportation. So I mentioned them. We have 3 major types of drug transport system. We have passive transport, we have active transport, and we have bulk transport. Ladies and gentlemen, let me tell you, if you are not here and you are missing some of these things, you are on your own. Because we are trying to make the things as simple as it is. So we have, for the passive transport is divided into 3. We have simple diffusion, we have facilitated diffusion, and we have osmosis. So for the simple diffusion, it's a transportation of smaller molecules and lipid-soluble substances. So any substance that is lipid-soluble or the molecules, they are very small, with just a simple diffusion, they can pass across the cell membrane. And of course, remember, diffusion is when there is movement of molecules from the region where their concentration is high to the region where the concentration is low. So that is why, because drugs are lipid soluble, the drugs, most of the drugs that are produced by pharmaceutical industry, they are lipid soluble. They are lipid soluble. And don't forget, ladies and gentlemen, the cell membrane is called lipid soluble because it is made up of lipids, that is lipid bilayer. I have a very comprehensive video on membrane structure. So generally, the lipid is made up of, the membrane of a cell is made up of a lipid. So that is why any lipid soluble substances can easily pass across the cell membrane. So the drugs that we are taking, the drugs that we are giving to your patients when they come to hospitals, they pass or they get into the cell to reach in the smooth endoplasmic reticulum through simple diffusion. And why is it that the drugs are passed across the cell membrane using this simple diffusion? Is because sometimes you need the drugs to reach into systematic circulation as soon as you take them. That is why you have even different route of drug administration. You have number one, it can be oral, intravenous, intramuscular, intradermal, because it depends on the situation of the patients and it depends on how you want the bioavailability of the drugs. So these are some of the important things that you need to know. And of course, for the facilitated diffusion is a type of transportation because the molecules are large or they are charged. So with simple diffusion, they cannot pass. So they need a channel. They need a channel for them to get into the cell. So that type of diffusion is called facilitated diffusion. Or sometimes they need a carrier, a molecules that can carry the molecules from outside the cell into the cell. So that's what happens in facilitated diffusion. So we have many of them, and we have osmosis, then for active transport. Of course, the simplest way to differentiate passive and active transport is that passive transport doesn't require energy to transport molecules, while active transport require energy for the transportation of molecules. And we have primary active transport. So the primary active transport is the types of transport that require ATP. And then, of course, for the secondary transport, it requires electromotive force or proton motive force to transport molecules. And then we have bulk transport, where we have endocytosis. So when you have a larger molecules like bacteria, so you need to apply endocytosis and exocytosis. Like, for example, during phagocytosis, you need endocytosis and exocytosis. So endocytosis is a type of transport in which a larger molecules like bacteria, like phagocytes, macrophages, dendritic cells or mast cells, identify a pathogen, identify a bacteria, engulf them, ingest them into the cell, and then they will be digested. So after digested, so the process in which immune cells grab and get the microorganisms into the cell is called endocytosis. While the process in which the digested products from the endocytosis, after the microorganism is digested, it breaks down, so they will get out of the cell. So you need exocytosis. So that is why, ladies and gentlemen, we do phagocytosis. I think it's what we even discussed last time in Human Physiology 225. I think we briefly discussed about phagocytosis, the steps of the phagocytosis. You can go and watch the video so that you can see how you can apply the knowledge of endocytosis and exocytosis in terms of phagocytosis of microorganisms. Please, are you following, ladies and gentlemen? So this is just a brief description of the transport of substances across the cell membrane. So then the next thing is lysosome. So what is the importance of lysosomes in a cell? Please, I want you to answer this question for you. What is the functions of lysosome in a cell? Please write it on the comment box. I'm coming. Write the functions of lysosomes on the comment box. Okay, so let me see, removal of excess excretory product, number one, and then lysosome help in digestion of substances, e.g. protein, breaking down of macromolecules, they transport waste material to the plasma membrane for ejection out of the cell. They produce digestive enzymes. It functions at digestive system of the cells. Lysosomes help in the degradation of cellular debris. So I think, ladies and gentlemen, you are all on point. What can I add explanation? Can I add explanation? All your point is correct. So if you said that, you'll have your full mark. But can I add explanation? Okay. So the lysosome is one of the organelles of the cell that contain an enzyme called lysozyme. That is the name of the enzyme. The content lysozyme. Lysozyme. So what is the importance of this lysozyme? Remember, I just actually finished explaining phagocytosis. So let's say this is a cell. You have a cell like this. Let's say this is macrophage as a cell, and this is your cellular debris. So when a macrophages come in contact with microorganisms, it will identify them and involve the microorganisms, and then the microorganisms will get into the lysosome. It will go into the lysosome of the cell. And then the lysosomes will now use these enzymes to degrade the bacteria or the microorganism. So you see, this is the bacteria. So you have the bacteria. So the bacteria will get into the cell through the process called endocytosis. So you have the bacteria, it gets into the cell through endocytosis, and then the bacteria will now move into the lysosome. And then the lysosome, of course, I think if you could remember from our last class, the physiology and biochemistry, they are actually related, especially in some area. So that is why if you have a good knowledge of biochemistry, you will definitely have good knowledge of physiology. So what happens actually, ladies and gentlemen, the phagocyte will fuse itself with the lysosome to form what you call phagolysosome. Phagolysosome. So when it fuses itself, you have phagolysosome, and then the phagolysosome fuse themselves together, and then there will be a secretion of the lysozyme, and then the lysozyme will now degrade this microorganism. So after degrading the microorganism, so the products of the digested product of these microorganisms will get themselves out of the cell using the process called lysosomes. That is basically transport. So ladies and gentlemen, I think you now understand the function. So the function of lysozyme is to degrade waste products or to degrade foreign microorganisms and then eliminate them, move them out from the cell for the body to be safe. So then the next thing is lysosome. Sorry, ribosome. So someone actually mentioned the functions of lysosome. Sorry, ribosome. So the ribosomes is also part of the cells that found in the cytoplasm. Remember, ladies and gentlemen, when we are talking about ribosome, so seriously speaking, or typically, strictly speaking, ribosomes is not an organism of the cell. Rather, it's falls under supramolecular complexes because the ribosome is formed by protein and ribosomal RNA. So we have ribosomal protein. So ribosomal protein together with RNA is what actually form ribosome. So that is why, strictly speaking, ribosomes is like also a supramolecular complex. So it's not a complete organism of the cell. So it's one of the supramolecular complexes. And remember, ladies and gentlemen, part of the discussion that we did on the organization of the cell or the life itself or all living organisms are started from atoms. So ribosomes is one of the supramolecular complexes. Apart from ribosomes, we have cytoskeleton, viruses, and prions are all examples of supramolecular complexes. So ribosomes is found in the cytoplasm, and the major functions of the ribosomes is for protein synthesis. Protein synthesis. So it uses RNA as a template. It uses messenger RNA, mRNA, messenger RNA as a template for protein synthesis. So this is the major functions of ribosome. So it involves in protein synthesis. So ladies and gentlemen, Hawawa Abubakar Gaddafi, Oyinye Thomas, is it Ostalo, aka Pickup, Christiana Yanyolua. So you didn't actually join my YouTube. So that means that you are not watching my videos on human physiology and biostatistic. So I'm telling you, if you are missing my videos on biostatistic, you are seriously missing a lot. Because I know that if you can watch my videos on biostatistic, you will never have problem with the course. So if you didn't join as a member, try as much as possible to join. Don't allow yourself to miss the opportunity of pursuing your courses. I think in one day, if you can watch all my videos on biostatistics, you will not have problem with the course. So after the ribosomes, the next one is nucleus. So what is the functions of nucleus? The nucleus is a control center of the cell, where the genetic material, which is DNA and RNA, are found. So I think if you can set that on RNA, it is the control center. So all the activities of the cell is controlled by the nucleus. So I don't want us to take much time here. So let's look at also another question. So you see, ladies and gentlemen, we now answer question 1A and 1B. So now question 1C. State the types of transport system involved in each of the following. Sodium glucose transporter, sodium potassium transporter, sodium potassium ATPase, release of insulin from pancreatic cells. So these are the different types of transport. These are like a kind of transportation across the cell membrane. So we have we have sodium glucose transport, sodium potassium transporter, then we have sodium potassium ATPase, then we also have release of insulin from the pancreatic cells. So please, ladies and gentlemen, I want to ask this question. Which types of transport system do you think it suits sodium glucose transporter? Please, I need answer to this. I need answer to this. Which types of transport do you think it suits? Okay, now can you see me again? Okay, let me share the question again. Can you see me on the question again? Okay. So someone said for sodium glucose transporter, someone said active transport. Someone said active transport. Do you agree? Sodium glucose transporter? Please, I need answer. Yes? Active transport? No. So seriously speaking, it's not an active transport. Remember here I said we have passivated diffusion. So passivated diffusion is for we have antipod, we have sympod and unipod. So you see when you have two molecules, let me actually you see I talk about the different type of transport where we have antipod, we have sympod, and we have unipod. So a sympod is when you have two molecules. Is when you have two molecules. Yes, it's passivated diffusion, but specifically, which type of the passivated diffusion? Is it antipod, is it sympod, or unipod? So when you have two molecules moving into the cell, moving across the cell membrane, at the same time when they are moving simultaneously into the cell, like glucose and sodium, glucose alone, or sodium alone, that is sodium ion, cannot move into the cell alone. Glucose also cannot move into the cell alone. Rather, it needs. Please, are you seeing me? I saw someone is saying that it is dark. Can you see me and also what I'm explaining? So what I'm saying, glucose cannot move into the cell alone. Sodium cannot move into the cell membrane alone. Rather, they move simultaneously into the cell. They move simultaneously into the cell. So that type of transport in which two molecules move into the cell simultaneously is called sympod transport. It is called sympod transport. So the answer to that question is sympod. So sodium, potassium, let me share the question again. So please, can you see the question? The question says briefly, okay, States the type of transport system involved in each of the following. Sodium glucose transporter, so the answer is SYMPOT. While for the sodium potassium transporter, sodium and potassium, there are two opposing molecules. There are molecules. Please, sir, I didn't get the pronunciation. The pronunciation is SYMPOT. SYMPOT. Look at it. SYMPOT. While in the case of sodium ion, potassium ion transporter, it is types of transport in which the two molecules moving in opposite direction. Where as the sodium is getting into the cell, then the direction. Sodium getting in and potassium getting out. So this type of transport is called antipod. So for question 1, you have, sorry, for Roman people 1, you have SYMPOT. For Roman people 2, that is sodium potassium transporter, you have antipod. So then the next one is for, okay, let me share it again, the Roman figure 3. So the Roman figure 3 is sodium potassium ATPase. So for the sodium potassium ATPase, from the word ATPase, it means that they require energy. So which type of transport require energy? Which types of transport require? So the types of transport between, sorry, in energy. So now transport require ATP, is active transport. But is it primary active transport or secondary active transport? That is the question. It is active transport, that is for the sodium potassium ATPase. But which types of transport? Is it active? Okay, you say it's active transport. So which of the active transport or secondary active transport? Excellent, is primary active transport. So the answer is primary active transport, not secondary. Because secondary active transport uses proton motive force or electrochemical gradient. So you see, we answered this question. So after this, okay, the next one is the transport of insulin. Let me share the last one so that you see the question. Last one, which is Roman figure 4. The release of insulin from the pancreatic cells. You know that generally pancreas produce insulin. And of course, one very important thing, excellent, the answer is exocytosis. Because insulin is a large molecule, it's a large protein. So therefore, it moves out of the pancreatic cells using exocytosis. So the answer to Roman figure 4 is. So, ladies and gentlemen, we now answer question 1 and 2 with a very clear explanation. So then after the question 3, so I'm done with question 2. So let's move to question 3. 1, 2, so let's move to question 3. So please, can you? I actually differentiate primary. I said to transport they require ATP, while for the active transport, they all require ATP. But the ATP in terms of the primary, so the energy that is required, they all require energy. But the energy for the primary active transport is ATP, while the energy in terms of the secondary active transport is in form of electrochemical gradient. So it's in the form of as proton motive force. So now we are moving to question 3. So you see, we answer question 1 correctly, and then we answer question 2. So now, ladies and gentlemen, I think from the last question, from our last class, I clearly explained. You see, 3A says, state the mechanisms of action of each of the following. The first one, we have aspirin as a drug. We have penicillin as antibiotics, and we have disulfiram. So these are the 3 drugs. So please explain what are the mechanisms of action of aspirin. So tell me which enzymes is targeted by aspirin. Please, I'm waiting for you. Aspirin, what is the mechanisms of action of aspirin? Excellent, excellent. Cyclooxygenase. You answer the question perfectly. It's cyclooxygenase. So please, ladies and gentlemen, if you want to really see or watch and get the explanation of mechanisms of action of aspirin, go and watch my last video on biochemistry. So we answered the mechanisms of action of aspirin and also that of the penicillin. So you don't need to waste time since we have already answered that, and I think you can actually go and check unless if you have some issues. Excellent, Charity. It blocks the production of prostaglandin because prostaglandins are the molecules that cause headache. Excellent. Then what about penicillin? What is the mechanisms of action of penicillin? What is the mechanisms of action of penicillin? Please answer the question. What is the mechanisms of action? Yes, Aisha Obala Babara, she said it inhibits bacterial cell wall synthesis. Yes, I agree. So it inhibits peptidoglycan biosynthesis. But what is the name of the enzymes that it inhibits? The name of the enzymes that it inhibits in peptidoglycan biosynthesis. Please, can you mention the enzyme? Trans 50 Ds. Yeah. So the enzymes that is blocked by this penicillin is transpeptidase. Is transpeptidase enzyme. So this is one of the enzymes that involve in the synthesis of peptidoglycan. So the penicillin is a drug that inhibits the activity of this transpeptidase. So therefore, peptidoglycan cannot be synthesized. So therefore, there will be a problem in terms of the synthesis of the peptidoglycan. Sorry, in terms of the synthesis of cell wall. So therefore, the microorganisms will be very fragile, so that our immune system can easily recognize them and kill them. So I think, ladies and gentlemen, you can clearly now understand how the mechanisms of action of these two drugs. Aspirin blocks cyclooxygenase activity to prevent or to inhibit the productions of prostaglandin, which linked to headache. And then second, the penicillin is an enzyme that blocks transpeptidase enzyme. So if you need more explanation on this, ladies and gentlemen, I advise you to go and watch the video. Yeah, the disulfiram is like anti-alcoholic drugs. It is a drug that if you are taking it, it's trying to prevent someone from taking drugs. Because at the end, this disulfiram, it blocks the activity of aldehyde dehydrogenase enzyme. It blocks the activity of aldehydes dehydrogenase enzyme. So I think so. As I was saying, the disulfiram is an enzyme that blocks the activity of alcohol, or that blocks the activity of enzymes called aldehyde dehydrogenase. So it prevents actually the oxidation of alcohol at the acetaldehyde level during alcohol metabolism. So, ladies and gentlemen, you need to understand how alcohol is metabolized in our body. So, when we take alcohol, what happens to it? So, generally, ladies and gentlemen, alcohol it's actually having a structure like this, like ethanol. So this alcohol can actually be converted into acetaldehydes. Acetaldehydes from alcohol. And the name of the enzymes that convert ethanol or the alcohol here to acetaldehyde is called alcohol dehydrogenase. Alcohol dehydrogenase enzyme. That is the name of the enzyme. So then the next enzymes that convert the ethanol or the alcohol, using alcohol dehydrogenase to acetaldehydes, is aldehyde dehydrogenase. Or you can now, aldehyde dehydrogenase. So aldehydes, aldehydes, dehydrogenase enzymes is an enzymes that involve in the conversion of acetaldehydes to ethanoic acids. So when you block the activity of this aldehyde dehydrogenase using disulfiram, it means that the acetaldehyde cannot be converted into ethanoic acids at this point. So if it cannot be converted into ethanoic acid, it means that the acetaldehyde will be accumulated. So when this acetaldehyde is accumulated, so it means that the alcohol cannot, you know that one of the functions of alcohol in our body, it produces energy. Because the alcohol, it involves in actually a reversible reaction. So an alcohol can be converted to pyruvate. But the problem is that you don't have knowledge of metabolism. But in your second semester, even right now on my YouTube channel, I have a video on the alcohol metabolism as well as the carbohydrate metabolism. But you are going to do it second semester. So that is what I want you to do, ladies and gentlemen. For you to have a very good grades for your second semester courses, immediately after you finish this examination, maybe you will take a 1 week break. So after taking 1 week break, so you are going to take like 3 or 4 courses for the second semester, where we are going to be discussing them at least time to time. So that at least when you resume the lectures, you will have a good understanding of the 4 courses. That is what we are going to target. So I have a lot of videos on those second semester courses, but it will be the best if you are willing and you wish to learn and you want to pass. And especially the second semester biochemistry and the physiology courses are much more difficult than the ones that you are doing now. So this 241 is simple compared to 242 that we are going to do second semester. So that's why we are going to start it at the right time. You see now, I'm still doing this biochem 242 to second semester student, but I'm not actually as the 22, rather we are doing it as a tutorial like as I used to do for your human physiology. So the problem is that I noticed that some of these courses, if you didn't start reading them on time, they will give you a headache. You will end up confusing yourself. So that is what these disulfuramides inhibit. So now I'm thinking, because we spent almost. So the next one, which is question 3b, differentiates the 3 types of enzymes inhibitors. Differentiate the 3 different types of reversible enzymes inhibition. Reversible enzymes inhibition. So please, ladies and gentlemen, I think you watch my video on the enzymes inhibition, on enzymology. And that is actually the end of our lecture, and you don't know that. So that is the end of our lectures. So I have already finished and I'm done with you regarding the Biochem 241. So we covered everything, we complete everything. I hope you are following. Simple format of the answer, please. What do you mean with the simple format of the answer? What do you mean with the simple format of the answer? So actually in this video, I'm going to actually stop this lecture because we spent almost 2 hours. So after I answer question 3b, so that is where we are going to stop. So we'll fix another time and answer question 3b. You see, ladies and gentlemen, I think I have 5 to 6 videos on revision to some of this question paper. So I usually don't answer the questions that very fast. So I usually follow the questions gradually for you to have a good understanding. The reason is because understanding is key, and also answering the questions at the same time is key. So sometimes the questions cannot be repeated. So it is very important to have a hint. If the question is not repeated, how can you approach and answer the other questions? So I have a lot of videos where I solve some of these past question papers. So I advise you to go back to my YouTube channel, especially the sessions. You will see most of those videos. So watch them. Just watch them. You will really enjoy them. So now, what are the different, because the enzymes inhibition, we have two types of enzymes inhibition. We have reversible enzymes inhibition and we have irreversible enzymes inhibition. So for the irreversible enzymes inhibition, for the irreversible enzyme inhibition, sometimes we call it suicide inhibition. Why is it called irreversible or suicide inhibition? It's because in the case of the irreversible enzyme inhibition, once the inhibition binds to the active site of the enzymes, so the binding is permanent. So the enzymes will be permanently inhibited. The enzymes will be permanently become inactive. So that is what happened in the irreversible inhibition. So it is suicide. So therefore, what I want you to understand in the case of irreversible enzymes inhibition, that type of inhibition is very dangerous because when we use the inhibitors at that point, the enzymes will become inactive permanently. But most of the drugs that are produced, they are not irreversible enzyme inhibitors. Rather, they are reversible enzyme inhibitors. So these are the two types of enzymes inhibition. We have irreversible and we have reversible enzyme inhibition. So in the case of reversible enzyme inhibition, it means that when you inhibit the enzymes, after removing the inhibitors, the activity of the enzymes will still maintain. So that's why it is reversible. You inhibit the enzymes, so after you remove the inhibitors, then the activity of the enzymes will still restore. So for this irreversible enzyme inhibition, we have different types. For this reversible enzyme inhibition, we have different types of them. We have number one, we have competitive inhibition, competitive reversible inhibitors. Number two, we have uncompetitive enzymes inhibition. And then we have mixed competitive inhibition. So these are the three types of inhibition. So in the case of competitive inhibition, is when the inhibitor and the substrate are competing with the same active site of the enzyme. Remember, when you have an enzymes usually have an active site. The active site of the enzyme is made up of binding sites, catalytic site, and also there are amino acid residues in the active sites. So what I want you to understand, ladies and gentlemen, an active site, it is a region of the enzymes where the substrate bind to. So this is an enzyme and this is your substrate. So in a situation where you have the inhibitor and the substrate have the same orientation, have the same shape, with the inhibitors and the substrate have the same shape, it means that the inhibitor at the same time can bind to the active site of the enzymes. Because, like, for example, look at it in this situation. This is the active site of the enzyme. So it can be, like in the case, it can be this one, can be the substrate. So you see the substrate and the inhibitor, they have the same shape. So it means that the inhibitor can bind to the active sites, and the substrate can also bind to the active sites. So in a situation where the inhibitor bind to the active site of the enzymes, that is what we call competitive inhibitors. So it is called competitive because the substrate and the inhibitors are competing with the same active sites. So in a situation where you have both the substrate and you have the inhibitor trying to bind to the active site, which one will bind? So at that stage, it depends on the concentration of the two. The concentration, or that is the amount of the substrate and the amount of the inhibitor. So if the amount of the inhibitor is higher than the amount of the substrate, so the inhibitor will bind. But if the amount, that is the concentration of the inhibitor, is higher than the amount of the, so the amount of the substrate is higher than the amount of the inhibitor, then the substrate will bind. So that is what happened in the case of competitive inhibitors. They are competing for the same active site. And then the next one is uncompetitive inhibitor. So the word uncompetitive, it means that they are inhibitors, but they are not competing with the active site. Rather, they are binding to other sites. So some enzymes can have another site in the enzymes apart from the active site. So in a situation where you have an inhibitor binding to other site of the enzymes, not the active site, so that type of inhibition is called uncompetitive inhibitor. And then in a situation where the inhibitors can bind with either the active site or the other site of the enzymes, that is what we call mixed competitive reversible inhibitor. So it means that in the mixed competitive inhibitions, the inhibition is when you have both competitive and uncompetitive inhibition happen or taking place in the same enzymes. That is what we call mixed competitive inhibitors. So, ladies and gentlemen, these are the three different types of reversible enzyme inhibitors. We have competitive, uncompetitive, and we have mixed competitive inhibition. So, ladies and gentlemen, it is actually almost time to pray. So, I think we spent 1 hour 15 minutes for this class. Please, I advise you, if you didn't join the YouTube community, try to join so that you can have access to our physiology videos, you can have access to our biostatistic videos. And I have group, WhatsApp group where we are doing the tutorial. So most of the students that are in that BNS tutorial group, the group will remain up to your graduation level. So we'll be using the group for your second semester courses. So if you are not in that group, so please, you can talk to me on my DM. I will send you the link to the group. And we'll get another time to also complete the solution to some of these questions. So, ladies and gentlemen, thank you very much. I hope you enjoy the discussion. So, what I want you to understand, ladies and gentlemen, please, it is now, you are now close to exam. So, have time to read. Have time to read. You see, that is why last time a lot of students are happy. They are happy with what I taught them. They passed their courses, especially the biochemistry and biostatistics. So last year I didn't do human physiology. So this is the first time that I started human physiology. So, and the video are also very interesting. And most of the students that joined us last semester, they are happy with what we taught them. So that is why, thank you, sir. Since I have been following you, I don't have fear for biochemistry again. Lucia, so thank you very much. So That is what I want you to understand. So we are going to stop here. I wanted to go on free. So thank you. See you next time. Lisa, I didn't understand. The mixed competitive inhibitors is when you have both competitive and uncompetitive. That is when the inhibitors combine to active sites and the other side of the enzyme. So thank you very much. See you next time.